Welcome to our course on neurophysiology and neurochemistry. In this part, you will learn something about the neurotransmitter dopamine. My name is Andreas Reif. I am a member of the ECNP Workshop Committee and head of the Scientific Program Committee from 2019 to 2021. Dopamine belongs to the class of monoaminergic neurotransmitters, which means that it is a biological amine formed by the decarboxylation of an amino acid. Also, it is a catecholamine. That means it is a biological amine which is also formed in the adrenal glands, which is the meaning of the Greek term catechol. Dopamine has important roles in the CNS, but also in the periphery, for example, regarding the tone of blood vessels. Dopamine was synthesized the first time at the beginning of the 20th century. However, it was discovered in the human brain as late as 1957. Soon thereafter, it was described as a neurotransmitter by the Swedish scientist Arvid Carlsson, a discovery which was awarded with the Nobel Prize for Medicine and Physiology in 2000. The synthesis of dopamine begins with the essential amino acid phenylalanine, which is converted to tyrosine by PAH, forming the dopamine precursor L-DOPA. This is the rate-limiting step in synthesis. DOPA is actually an abbreviation which gave rise to the name dopamine, the amine which is derived from DOPA. L-DOPA, which can cross the blood-brain barrier, is then decarboxylized to dopamine in neurons. There are also other, however minor, synthesis pathways. Catabolism of dopamine is manifold, with three relevant pathways. Dopamine can be hydroxylazed by dopamine beta-hydroxylase to norepinephrine, which is noradrenaline, and this is a neurotransmitter itself. Or it can be turned into 3-MT by COMT, or it can be converted to DOPAC by the isoforms of monoamine oxidases MAO-A and MAO-B in sequence with ALDH. MAO and ALDH can turn 3-MT into the main breakdown product homovalenic acid HVA, while DOPAC is metabolized to HVA by COMT. Importantly, many of these enzymes are drug targets, which will be seen in one of the subsequent slides. Dopamine signaling is very complex and features several postsynaptic mechanisms, which are different in distinct brain regions or between neuronal cell types. There are two main classes of dopamine receptors: the D1-like class, comprising of the D1 and D5 receptor, and the D2-like class, which is formed by D2, D3, and D4 receptors. D1-like receptors are exclusively postsynaptic, while D2-like receptors can be found at pre- and the postsynapse. The main difference between both classes is their coupled G protein. While D1-like receptors are coupled to stimulatory G proteins, D2-like receptors bind to inhibitory G proteins. Most interestingly, D2 receptors occur in two varieties, so-called isoforms, a short one and a long one, which has implications for the binding rate of inhibitors. Dopamine reuptake can be accomplished by the postsynaptic cell by the dopamine transporter DAT. In the presynaptic neuron, dopamine can either be degraded by COMT, the predominant way in the prefrontal cortex, or MAO, or it can be stored in vesicles. The vesicular monoamine transporter VMAT2 is the molecule that transports dopamine into the vesicle. Binding of dopamine to its receptors triggers a plethora of intracellular mechanisms. The first step is the activation of G proteins, activating ones in the case of D1-like receptors, inhibitory ones for D2-like receptors. G proteins can then either activate or inhibit an enzyme called adenylate cyclase, a key regulator of second messengers. Adenylate cyclase forms cyclic AMP from ATP, and cyclic AMP in turn activates protein kinase A. This enzyme phosphorylates other proteins in the neuron and thereby orchestrates numerous other pathways. Another central molecule is STARP32, which is phosphorylated by protein kinase A and which is inhibiting a dephosphorylating protein called PB1. Ultimately, dopamine receptor activation results in an increase or decrease of neuronal excitability by affecting potassium and calcium channels. D1 and D2 receptors can also dimerize and form heteromers. Their activation then triggers phospholipase C activation, resulting in the formation of IP3 and subsequent release of calcium from the endoplasmatic reticulum. As you can see here, there are 4 major dopaminergic pathways in the human brain, actually 5 when you also consider intrathalamic pathways. The smallest pathway is the tubero-infundibular pathway originating in the hypothalamus, which regulates prolactin secretion from the pituitary gland. The nigrostriatal pathway originates in the brainstem nucleus substantia nigra and projects to the basal ganglia, more specifically to the striatum. This pathway is part of the extrapyramidal system, in turn part of a motor system network causing involuntary actions. By this pathway, dopamine has an important role in movement disorders. The mesolimbic and mesocortical pathways are the ones which are of special interest in psychiatry. Both originate from the ventral tegmental area, the VTA. The mesolimbic system projects mainly to the nucleus accumbens and is involved in reward and motivation, which is relevant for schizophrenia and depression. The mesocortical pathway projects to the prefrontal and frontal cortex. Together, the mesocortico-limbic projections play a role in ADHD, schizophrenia, and substance abuse. As dopamine is critically involved in the regulation of several brain circuits, unsurprisingly, it has an important role in several disorders. First, degeneration of substantia nigra neurons leads to Parkinson's disease, which is characterized by rigor, tremor, and akinesia. Dopamine, however, also contributes to other movement disorders, such as choreatic movements in Huntington's disease, restless leg syndrome, and especially inherited dystonia, which is dopa-sensitive. Dopamine has an important role in reward processing, or more specifically, the prediction of reward, as shown in this classical experiment by Schultz. In case of an unexpected reward, dopamine neurons in the striatum will fire, while they show dampened activity when reward is expected but not coming. This essential mechanism implicates dopamine signaling in all psychiatric disorders that feature abnormalities in reward processing. These include depression and anhedonia, also in the context of other mental disorders. On the other hand, dopamine is involved in disorders where reward too soon wanes off, which can be found in excessive sensation seeking as a trait or in the disorders ADHD and substance use. Some drugs of abuse, such as amphetamine, also directly stimulate the dopamine pathway underlying their abuse potential. Finally, dopamine is crucially involved in schizophrenia. There, excessive dopamine neurotransmission is thought to underlie so-called positive symptoms such as delusions and hallucinations. Psychopharmacology is rich of compounds manipulating the dopamine pathway. Dopamine agonists are mainly used in Parkinson's disease, but also restless leg syndrome and endocrine states going along with increased prolactin. The most common agonist is L-Dopa, the precursor of dopamine, and this can cross the blood-brain barrier and is there converted to dopamine. More specifically, there are receptor agonists mainly targeting the D2 and to a lesser extent D3 receptors. Blockade of the D2 receptor is thought to be the main mechanism of action of first-generation antipsychotics, the most prominent one being haloperidol. Some second-generation antipsychotics also display by blockade of the D3 receptor, which is thought to contribute to antipsychotic efficacy. Dopamine metabolism is also a major target for drugs. In Parkinson's disease, one tries to increase dopamine levels, and this is done via blockade of its catabolic enzymes COMT and MAO. MAO inhibition is also used in depression. However, there it may be also important for serotonin metabolism, and this is also broken down by MAO. Furthermore, there's a niche indication for VMAT2 inhibition. Finally, blockade of the dopamine transporter DAT is used as a therapeutic principle in ADHD, where methylphenidate is a potent DAT and lesser so NET inhibitor. Amphetamine even reverses dopamine transport across the membrane and causes release of dopamine from synaptic vesicles. In doing so, it massively increases dopamine levels in the synaptic cleft. Taken together, the dopamine system has many important roles in psychological and psychomotor approach behaviors and is involved in the pathogenesis of many neuropsychiatric disorders. Dopamine pathways are a target for numerous medicinal and illicit drugs. Both basic as well as clinical researchers should therefore be well acquainted with this fascinating molecule. Thank you for listening.